Hello, Standard 9, going to teach Chapter 9, Economic Importance of Bacteria and Fungi, and it is Part 4. Dear children, we have already discussed the role of bacteria in medicine. That part we have discussed. Now we are going to discuss role of bacteria in industries. So, where it is used in industries? Number one, it is used in flavoring of tea. Flavoring of tea. And next, it is used in tanning of leather. Now, flavoring of tea and tanning of leather, let us discuss in detail. When the tea leaves are plucked, it is green, it is dried and left for some time, and the flavor which comes in the tea, that is due to the bacteria only, and that process is called as tea curing. That process is called as curing of tea or tea curing, and due to tea curing only, the flavor is there in the tea. So that part is clear. In the tanning of leather, listen, when hides or skins of animals are taken that contain blood vessels, hairs, many unwanted things are there. Generally we dry it and that skin is made to leather. But there are certain bacteria which removes the unwanted substance from the skin, and the skin is changed to leather, and that process is called as tanning of leather. So role of bacteria in industries in the flavoring of tea and in the tanning of leather. Now it was about the beneficial effect of bacteria. Let us discuss some harmful effect of bacteria. Okay. In harmful effect of bacteria, spoilage of food. Spoilage of food. Food is spoiled due to bacteria only. Okay. Especially during the summer season, cooked food, milk, fruits, vegetable, they decay, they are fermented, and that is due to the bacteria only. And sometimes it may lead to food poisoning and botulism. Botulism is a type of serious food poisoning. Okay, very serious food poisoning due to the special bacterium. Okay, sometimes found in tinned and sealed foods, especially if we will use tinned and sealed food. In that, there may be case of botulism, a serious type of food poisoning. Okay. So that part is clear. Now, the affected canes get distended at their end with gas, which gases out open when we are opening the tinned or sealed foods. Gases gusses out, and that will lead to the bacterial infection. Now, that part is clear. Botulism is most important for the examination point of view. Let us discuss how we can preserve our food. Food preservation. Okay. First method, boiling or heating. Preservation of food. By boiling or heating. But one thing, Water boils at 100 degrees Celsius. At 100 degrees Celsius, spores are not killed. Spores of bacteria are not killed. Okay. Then we have to raise the temperature till 110 degrees Celsius. But how we can raise the temperature? Because if we will provide temperature, the water will boil and the temperature of the water will not rise above 100 degrees Celsius. Then we have to use autoclaves, or we can use pressure cooker. And in autoclave and pressure cooker, the temperature can be raised, and there the spores will be killed. And that is one of the way for the food preservation. Okay. Now salting. Next method is the salting. You have seen that pickles contain more amount of salt, or there are salted fish, salted meat. Okay. So second method is the salting, means addition of salt. And if in cheese or many other food item, if we will add salt, bacteria will not grow there. Okay. And treatment of salt for preservation is also called curing. Okay. Treatment with salt is called as curing. Next is dehydration. Means removing water. How water is removed? By drying. You have seen your mother dry the food item. Okay. And in that, in less amount of moisture, the bacteria will not grow. Nowadays, means milk is also dried and made to powder, and milk powder can be preserved for a long period of time. Next is irradiation. Let us discuss about the irradiation. So what is irradiation? Irradiation means radioactive radiation. Okay, and radioactive radiation kills the germs. Nowadays, ultraviolet light is also used. UV light is also used to kill the germs. And this way, if the microbes are killed, the food can be preserved for a long period of time. Next is pasteurization, and pasteurization is very, very important for the examination point of view. Pasteurization. So, what is pasteurization? It is done to the milk. So, what is pasteurization? Heating at 60 degrees Celsius for 30 minutes, half an hour, 30 minutes. If we will heat the milk at 60 degrees Celsius for half an hour and chilled and packed, then it can be preserved. But pasteurized milk, it doesn't mean that all the bacteria are killed. No, some spores of bacteria are already present there in the pasteurized milk. So pasteurized milk must be kept in refrigerator or at low temperature so that the spores will not grow, and it can be preserved for a long period of time. And since it was developed by a scientist named Louis Pasteur, so that method is known as pasteurization or pasteurization of milk. Okay. Next, refrigeration. In refrigerator, the food items can be kept fresh for a long period of time. In our fridge, the temperature may go up to -5 degrees Celsius. But in deep fridge, or in commercial phrase, the temperature can go up to minus 20 degrees Celsius to minus 30 degrees Celsius. And there in deep fridge for a long period of time, the food items can be kept. Now, a strong next method, strong concentration of chemical. A strong concentration or use of chemical can be there for food preservation. Okay, a strong concentration means if sugar solution, a strong sugar solution is there and the food is kept in the sugar solution, it will remain fresh for a long period of time. And in jams, you have seen jams, jellies, they are preserved for a long period of time. And some what will happen if the concentration is strong, bacteria will not grow there. Plasmolysis means bacterial sap will be taken out and it will die. Okay, next thing, chemical, one chemical, sodium benzoate. Okay, sodium benzoate can be used as food preservative for a long, to keep food fresh for a long, and it is used in jams and jellies, sodium benzoate. So these are the methods by which we can preserve our food. Now some more thing which we have to discuss, and that is bacterial disease in plants and animals. Bacterial diseases in plants and animals. Now we will discuss some of the diseases of the plant which are caused by bacteria and some of the diseases of the animals which are caused by bacteria. In plant, black rot of mustard. Black rot of mustard. Okay, black rot of mustard and cauliflower. And cauliflower is caused by the bacteria. And one more disease, and that disease is, this disease affects mustard and cauliflower. And other diseases, bacterial blight of cowpea. Bacterial blight of cowpea. Cowpea means lobia. Okay, that is caused by the bacteria and the production is reduced. So it was about the disease in plants. Now let us discuss bacterial disease in animal. One of the important bacterial disease in animal is the anthrax. Okay, anthrax, common disease of animals. Okay, the milk production is reduced, the body swells. Okay, swelling on the body, and milk production is reduced. Okay, now next is bovine tuberculosis. A type of tuberculosis, bovine tuberculosis, also affect the milk production, and lungs are affected. The animal's lungs are affected, and husky cough, sukha khasi, is caused in bovine tuberculosis. Now in humans we have already discussed that tuberculosis, whooping cough, cholera, diphtheria, typhoid, pneumonia are caused by bacteria, which was already taught in the chapter diseases. Now one very important thing which we have to discuss, and that is bioweapons. Okay, bioweapons. Nowadays, what is done? Such type of bacteria and viruses are developed in laboratory and they are released in the atmosphere. For example, if tetany bacteria is developed and it is released, maximum will suffer from the tetanus. as a bomb it can be used. And nowadays, whatever you are, we are suffering corona. One of the thesis behind it that this coronavirus is developed in the. It is said, I can't say confirmly, that it is said that it is developed in the laboratory of China, and by mistake or accidentally it came out, and we all, whole world is suffering from the corona disease. And that is, corona is used as what? Used as bioweapons. Okay. So that virus COVID-19, that is also used as bioweapon, and very dangerous, effects almost whole world. So it was about the bacteria. So dear children, we have already discussed about the harmful and useful role of bacteria. In the next video, we will discuss the rest part. Thank you very much.